Transcription and translation are the two stages of protein synthesis. The simplest framing. Transcription copies DNA into mRNA. Translation reads mRNA to build a protein. The central dogma flows DNA to RNA to protein. Transcription comes first. Translation comes second. Transcription happens in the nucleus in eukaryotes. The enzyme RNA polymerase binds the promoter, unwinds the DNA, and reads the template strand from 3' to 5'. It builds mRNA from 5' to 3'. The key swap. Uracil replaces thymine in RNA. So DNAA becomes mRNAU. When polymerase hits a terminator, the mRNA is released. Translation happens in the cytoplasm at ribosomes. The ribosome binds mRNA at the start codon AUG, which always codes for methionine. tRNA molecules deliver amino acids. Each tRNA anticodon pairs with an mRNA codon. Peptide bonds form between amino acids. The ribosome moves along the mRNA 3 nucleotides at a time. When it hits a stop codon, UAA, UAG, or UGA, release factors free the polypeptide. 3 different processes graders love to confuse. Replication copies the entire genome from DNA to DNA using DNA polymerase. Transcription copies one gene from DNA to mRNA using RNA polymerase. Translation reads mRNA to build a polypeptide using ribosomes and tRNA. Different templates, different machinery, different products. Memorize all 3 together. The location difference matters on the AP exam. Eukaryotes separate the 2 processes. Transcription in the nucleus, translation in the cytoplasm. Eukaryotic mRNA also gets processed. 5' cap, 3' poly A tail, tailatial, introns spliced out, exons joined. Prokaryotes have no nucleus, so transcription and translation happen at the same time in the cytoplasm. Bacteria can begin translating an mRNA while it's still being transcribed.
The link below has 22 flashcards and 16 AP-style practice questions on transcription versus translation. They're built to help you ace the AP exam. Try them. Subscribe for new AP videos every week.